Hi, I'm Doug Piper of Gourmet Brewing, and I really appreciate your interest in viewing this recent presentation from Richard Dubé of the Siebel Institute. If you like this video, please know that Gourmet Brewing's content is entirely crowd-supported, so please consider supporting future content by supporting us on our Patreon channel. I was fortunate to have the opportunity to record Richard's presentation during his November 2019 visit to the North Carolina Craft Brewers Guild annual conference. In this video, Richard covers many of his recommendations to measure and improve the microbiological activity in your brewery. So let's join Richard Dubé of the Siebel Institute during his presentation at the 2019 North Carolina Brewers Guild annual conference in Winston-Salem, North Carolina.
One of my favorite beer educators. Somebody that I get to work with quite a bit, so it's good. It's nice to be able to introduce him here. The presentation is going to be Micro What? Detecting and Controlling Yeast and Bacteria from Grain and Glass. Richard has a Bachelor of Science in Microbiology, Biochemistry, and earned an Associate's Diploma at the Institute of Brewing in London. First worked for Molson Breweries in quality control and research, then occupied various positions in all production departments with Labatt's and Budweiser, relocated to the U.S. to offer the responsibilities of product development and quality control in several contracting brewing sites under Boston Beer Company, partially retired, which he would have stayed retired a little bit longer, in 2000 and taught high school science for the next 10 years. Richard returned to the brewing industry in 2011, accepting the challenge of opening a large brew pub on the banks along the Ohio River in Cincinnati. In May 2014, he took a permanent position with the Siebel Institute of Technology as the e-learning education specialist, while also becoming a co-founder for the first freestanding microbrewery in northern Kentucky. So give it up, this is going to be a good talk. And
thank you very much. Can you hear me? Back there? Because I hate working with this, okay? Because I like to work around. So thank you very much for the introduction. Thank you very much. Thank you very much, everyone, for being here today. We'll try to look at a couple of basic micro stuff. Our time is limited, so I'll give you some tools to where to go to learn a little bit more, hopefully. So my title is Eliminate Contaminants. Don't kid yourself, that won't happen. So my content goes like this. I'm going to go through a couple of definitions just to refresh a couple of terms. Why bother? Why bother controlling microbes in our brewery? I'll give you some basic equipment that anybody that starts in the industry, even at the lower level, should have access to those basic piece of equipment. Where are they? So where do we go to start detecting those microorganisms? And I give you what I call my 8 steps of control by prevention, because again, don't imagine that you can eliminate, you can only control, and the best way to control is by prevention. And finally, there's a lecture for that, and I'll keep that for the end. So a couple of definitions. So when we're talking about beer spoilage, we're obviously talking about something that will change the appearance, not only the appearance of your product in terms of maybe haziness or turbidity or ropiness or anything like this, but obviously anything that could affect the flavor and the aroma of your product. So hopefully you know what your flavor profile of your beer is, and so therefore you can detect if it's getting off of that. Talking about obviously diacetyl, DMS, barnyard, whatever you want to put in this equation, but this is what we call beer spoilage, anything that will affect the appearance, flavor, and aroma of your product. Now with that in mind, so what is a brewing contaminant? Well, a brewing contaminant, in simple term, is anything that is not your culture yeast. Seems fairly straightforward, but with this definition, we can extend that, let's say your ale yeast, if you find your ale yeast in your lager yeast, this is a wild yeast, okay? And this is very important to understand. It is also important to understand that anything that is not there on purpose is obviously a brewing contaminant. And when we talk about brewing contaminant, we have basically two huge classes. We're talking about spoilers and non-spoilers. And in my book, they are as important one to the other. The beer spoiler is obviously what will cause those change of appearance and flavor and aroma. But the non-spoilers is something that cannot grow in your beer, but if it's in your environment, it will become a possible source of nutrient for your spoiler. So one and the other are as important, and they should be eliminated, or again, not eliminated, but at least controlled, okay? Okay, so basically when we're talking about brewing contaminants, we're talking about basically three things: molds, bacteria, and yeast. So quickly on molds, we're talking they are strictly aerobe, so they don't grow in your beer. So you say, okay, that's no problem. So I can let my white walls become black. Well, no, not really. Okay, this is not what you're supposed to do. You're supposed to eliminate those guys, because at the end of the day, they give you a reflection of the quality of the overall hygiene of your brewery. You should be able to eat on the floor. This is my approach. You should be able to eat on the floor of your brewery. If you can't do that, telling you, go back to square one and start cleaning out and scrubbing. They represent a health hazard, simply because they grow, they produce metabolites. Those metabolites find themselves in the environment. You can breed them, like, of course, it's not really for us brewers, but there's a famous disease, farmer's lung, that is crops that are infected with Fusarium, for instance, or Aspergillus niger, that actually is transported into the dust and goes into the lungs of the farmers, and then create a very damaging disease. But they also produce some toxins, and some of those toxins are not only carcinogenic, but they're very heat stable. So they can not only survive the malting process, but also the brewing process. So you need to eliminate those guys as well, or control them as much as possible. And then finally, the molds are spoilage without contact. Again, because they are strictly aerobe, they will not grow in your finished beer. But again, those metabolites, or those nice black mold growing on your walls, can find itself in the environment. And I've seen it in several breweries. You have a nice pallet of cans waiting to be filled the next day, and it's all open, unwrapped. So what do you think can happen? Well, those metabolites can find their way in the cans, and then you end up with moldy taste and earthy, moldy taste on your beer, and this has to be controlled as well. Finally, Fusarium is also known to be associated to gushing. Gushing, this uncontrollable reaction or foaming of your beer. It's at the right temperature, it has not been shaken, and then you open it and then it gushes like crazy. It is associated a lot with this mold Fusarium. It's a small peptide called hydrophobins that creates that reaction. The other group is, of course, the bacteria. We have 5 huge families. Here they are, anything and everything from lactic acid bacteria, we call them lab, lactobacillus, pediococcus, the enterobacters, and there goes the list, bacillus, bacillus thermophilus, that is really found mostly in the plate heat exchanger, and I'm going to mention that later. Pseudomonas and the acetic acid bacteria, the AAB, that is also kind of very interesting because they are mostly strictly aerobe, so they will not grow in what I call properly packaged beer. But it can definitely cause some issues, and I'm going to show you that later. And of course, we have our wild yeast, and I want to restate the definition. Wild yeast is anything that is not supposed to be there. So lager yeast and your ale yeast, or vice versa, that yeast is a wild yeast in that context. We have some Saccharomyces type wild yeast, and we have non-Saccharomyces type wild yeast. I'm talking about Brett, for instance, Brettanomyces. What's interesting about the wild yeast is to understand that just like bacteria and molds, they are ubiquitous, meaning that they are found everywhere. But the further problem with wild yeast is that because they go from aerobe to facultative anaerobe, meaning that they're very comfortable to grow with or without oxygen, so therefore you can understand that you can find them absolutely everywhere. They come in with your raw material, they come in water, they come in on your body, they come in from your bud, from your chum, from your friend that brought his homebrew and he wants you to analyze it and the bucket is all overfilling and everything. It comes from everywhere, so you have to think about this. And of course, you know, it causes the spoilage that we've mentioned before, biofilm, haze, over-attenuation like diastaticus. I'm going to talk about this monster that has been on the news for the past few years now, and that cause over-attenuation and some off-flavors. Okay, so why bother with microbiological control? Well, at the end of the day, it's very simple. You have to keep that under control. You have to keep anything and everything that is not your culture yeast, you have to keep that to a level that will not cause you some problems. I will state that, and I will say it several times, your brewery is a natural ecosystem. It has its own ecoflora. So don't think that you can sterilize your brewery. You can simply maintain a level of contaminant that is below what can cause some problem, okay? And those that are a little bit more involved in microbiology, by the way, when you see no growth on a plate, okay, the number to report is not 0, is ND, non-detectable. Because when you try to explain that to people that are just looking at your report, say, hey, there's nothing. We're good, we're clean, we're perfect. No. It's non-detectable. They are there. They're just at a level that you are controlling that will not cause you some problem. Maybe a little bit philosophical here, but it's very important if you really get into a huge microbiology program and you report zeros, this is wrong. This is not the way to go at it. Plus you have to specify the volume that you have inoculated, okay? Of course, if you have some issues and you have to recall some products, it's very, very expensive, and you can make a very, very quick calculation that to recall product is so much more expensive than having a very, very basic microbiology program. Here, just pictures of, does that work? It didn't seem much, but anyway, put Eucoccus over here, some lactobacillus, and some wild yeast on top. Okay, so where do I start? Well, you don't need a microbiology degree, and you don't need unlimited funds to have a basic microbiology program. Where you need to start, though, you need to start with a location. And it doesn't need to be huge. I remember when we first started, we had just a little room, 5 by 5, you know, but well organized, something that you can close, something that there's not a lot of air traffic or airflow. We had a sink, and it's an area that you can keep free of dust. So that's basically all you need to start with in terms of location. Now, what about some equipment? Oh, well, sorry, here. Very simple to keep a sterile space. You need a sterile space that is about 12 by 20 inches, and all you need is a porta burner or a Bunsen burner, and that will create an area, a sterile environment in front of that burner that is sufficient to do all your micro work. What else do we need? Well, we need a microscope. Okay, we need a microscope. The microscope is there to, of course, magnify what the eye can't see, and it's there to give you some differences to be able to differentiate, you know, between either some organelles or some condition of your yeast and that kind of stuff. You can buy a very, very good bright field microscope. It's basic. It's not very expensive. But the basis of a bright field microscope is that basically you have to use stain to be able to clearly see the organisms, because you end up with a very bright background with a little bit of darkness created by the interference of the organism itself. So you don't see a lot of detail, not a lot of contrast. Now the price have come down tremendously. Me, I recommend buy a phase contrast microscope. It gives you a lot of detail. It truly, as the name say, it gives you a tremendous amount of contrast. And when it comes down to looking at your yeast and getting familiar with your yeast, a phase contrast is much better. Okay, for about 800, last one I bought for the brewery, five years ago, it cost me less than $800. Okay, and it's really, it's maybe an extra $500 between the bright field and the phase contrast, but I'm telling you, it's much better. And a friend of mine that wrote a book, the Handbook of Brewing, you have to keep that in mind. Microscopic observation of brewery pitching yeast is as important today as it was when Pasteur first looked at it. Okay? You need to know your yeast. You need to look at your yeast all the time. And you need to have a mental picture of what my yeast looks like after pitching, after 24 hours in fermenting, at the end of fermentation, etc., etc. You have to have that. And nowadays it's so easy. You can have some adapter to put on your microscope and use your phone and take some pictures. Because remember, unless you want to work 365 days a year, you're probably going to need some replacement at one point because you take some vacation or you're sick or whatever, whatnot. And that person needs to be equipped to do the same job as you are. And a nice little digital album of pictures, with some notes goes a long way. Another list of micro-related equipment. I say incubators, don't break the bank. What you see there on the left-hand side is actually a reptile incubator. It's about 5 times cheaper than an incubator, but it does exactly the same job, okay? And below, it's a little bit difficult to see, but it's a DIY anaerobic incubator. It's just call it a Tupperware. Now the seals are excellent. A Tupperware to which I have adapted a small quick disconnect to that I use with a CO2 cylinder. Flush it a couple of times, seal it, and there it goes. You have an anaerobic incubator. A pH meter is critical, heat stirring plate, or maybe just a pressure cooker that you can find in your kitchen. A water bath, and I put that over there. It's just a sous vide cooking system for about $100. It's beautiful because it maintains the temperature. You can use it for your media. You can use it for force aging testing. You can use it for forced diacetyl at the end of fermentation. Cheap. Porta-gas burner, nitro gloves, steel tubes, Erlenmeyer flask, a scale that can measure to a hundredth of a gram, some reagent bottles, and finally some steel pipette so that you can just throw away or buy an auto pipetter with steel tips. Okay, so where the hell are they? Okay, where are they? Well, you can't sample everything. Okay? You cannot imagine that you can sample everything. The stupid analogy is to say, okay, I need to check every single cans that comes out of my can filler. Well, obviously, there's nothing left to be sold, so you're in trouble. Okay? So you have to understand your CCPs, your critical control points. Okay, what is critical not only in terms of overall process, but also in terms of your own facilities, okay? The layout. So you have to know the layout of your brewery and understand where it is that it's a little bit more critical to look into it. So here I just write, your raw materials, your cold wort, your yeast slurry, and again, specific location based on your own brewery. What is the baseline? And I call it the Polaroid, to just show my age a little bit here, Polaroid picture. Polaroid picture means this, okay? Before being able to get yourself into what I call an investigating mode, which is for the microbiologist or anybody who's going to be in the lab to do that, it's much more fun to investigate than to do thousands of freaking samples that you do every week. So what you do is that you take your entire brewery and you cut it in small pieces, let's say water system, brew house, wort cooling, fermentation, etc., etc. And then you see when everything goes well, everybody's happy, you know, the beer is great, you like it, but what is the microbiological baseline of what it is now? You take some samples and you see that, okay, yeah, this is what it looks like, and you establish again what I call a baseline against which you can compare some results. And then you apply some basic screening, okay? We're gonna see wort sample, yeast slurry, tanks, any tanks from your CCV to bright beer tanks, and the packaged product. When we look at the wort sample, the Death Star of the wort is obviously the plate heat exchanger. It can harbor some nasty bacteria like Bacillus thermophilus, but a lot of also Enterobacter. Those suckers doesn't grow in beer, but then can cause enough problem before, especially if you don't pitch properly and you end up with a longer lag phase. Well, remember that the importance of the lag phase is to kick up the fermentation very quickly. Because if you kind of wait, because for a while the yeast doesn't do anything than just getting some energy to be able to create more cellular component to have babies, to have daughters. But during that period, well, if you have a high level of contaminants, they can produce enough crap, lactic acid, DMS, and the like, that can stay there. They will not survive once finally the yeast have kicked off, but it has already caused some problems. So what to do? Very simple. Take a sterile tube. You know, those either you autoclave them or you buy them already pre-sterilized. Take a cold wort sample. Put it at room temperature, and I put room temperature in parentheses because if you're talking to someone that is in South Africa or if you're talking to someone that is Antarctica, well, room temperature might be different. So we're talking about, you know, 20 C's, 70 degrees Fahrenheit or something. Or put them on your incubator, or like I've seen, which I thought was a great idea, put them in your boiler room. Yeah, boiler room is always kind of warm enough. Just put them there. There goes your natural incubator. And what you're looking for, 24 hours. I'm telling you, if you have something, 24 hours, 48 up, you'll see it. If you're really paranoid, keep it another 24 for a total of 72. But you're going to see quickly a change of turbidity. You're going to see the formation of gas. You're going to look at it and you just shake it a little bit and you see that there's a little bit of gas that has been formed. And then you open it, or just when you open it, it's going to go psh, and you know that that's not right. And then you smell it and you're going to get the DMS parsnip type of character. And then if you have something and you can eliminate sampling error and what I call the cleaning in place procedure mishap, you know, something that you can recognize, well, it's time to open the plates, because your plate's going to look like this. Even if you do the perfect CIP after each of your brews in both directions to make sure, etc., eventually you're going to have to open your heat exchanger. Not fun, but it has to happen. Yeast slurry, well, direct microscopic observation, or DMO, is that my yeast? Learn to recognize your yeast. I mentioned it, but I'm going to say it again because it's important. You have 5 minutes, you have half an hour that, you know, everything seems to be quiet. You know what you do? You grab some yeast. Doesn't matter, to just keep track of what you're doing. You grab some yeast from a fermenter and you look at it. This is what it looks like when it's in full fermentation, 48 hours, it's active. This is what it looks like. Take a picture, take some notes. You have to be able to recognize what your yeast is supposed to look like when everybody's happy. Is there any contaminant? Especially in the yeast slurry, you're going to be able to see if you have contaminants. Then in terms of a couple of tests, viability, well, I think that everybody knows Maitland Blue or Maitland Violet. It's a little bit of a personal choice here because they do about the same thing. But you have to realize that it's totally unreliable when you get below 85%. If you get below 85%, you might as well be at 45%. You have no clue. Okay, so you have to be above 85% to say, okay, I'm in good shape, I can repitch. And when you repitch greater than 90% viable. Because the idea that I've heard that, well, I'm at 75, so you know what, I'm going to pitch 25% more. Well, what the heck do you think you're pitching? You're pitching dead yeast, okay? Dead yeast that open its innards, push all the garbage out, and this is what you end up with. And then you say, God, my beer is like pretty umami, kind of the broady taste. Well, where do you think it comes from? It comes from your yeast that you put extra just because you wanted to have a good fermentation. It doesn't work that way. That test, that pH test, I love it because it's very, very simple. When you collect your yeast, okay, and you should do that anyway, you do an analysis on the beer that it comes from. And you do a pH, and the pH is whatever, 4.2. So you have to agree that the slurry also is also at 4.2. And then you sit on it for a few days, and then you want to reuse it. Okay? Well, can I reuse it? Do a pH on your yeast slurry. If there's any increase of pH compared to the beer where it comes from, it gives you an indication that the yeast has autolyzed. If it's greater than 0.5 unit, probable. If it's greater than 1 unit, it's a certainty that the yeast has autolyzed, spills the innards, and increase the pH, so it's not something that you should reuse. I love this because it's very, very simple, straightforward. When we look at vitality, so viability is about the ability to reproduce, whereas the vitality is the ability to perform. Meaning that I'm still alive, but I'm not I cannot perform running as well as I was running when I was 20 years old, okay? So I'm alive, I'm here, but not as in good shape as I used to be. Whereas viability is that there's no gray zone. You're either dead or you're alive, okay? So when it comes to vitality, another very easy test. And again, it's for you to play with the baseline, play with it, and see where you land. But take your slurry. Most of you should do your iodine test in the brew house. Take the same iodine and apply it to your yeast slurry. If the yeast slurry is in good shape, it has a tremendous amount of glycogen reserves. And you're going to be able to detect it with iodine. It's going to turn brownish. The browner it is, the greater the glycogen reserve, the better your yeast is ready to rock and roll. Okay? Well, is it our ale or lager culture? Well, the direct microscopic observation, despite the fact that you know what it looks like, it's not an absolute tool or result to use. So there's some other things. Just plate it, and there's a tremendous, very, very clear difference between ale and lager yeast. Ale will be kind of rough and a little bit smaller colonies, and lagers would be a little bit larger and very smooth. And if you use a media like WLN that contains bromochrysal green, that is being absorbed by the yeast, and it's strain dependent, you're going to end up with different shade of green of your colonies, and that's another way to look at it. And another very easy way is that you plate something, put it at 37. If something grows, it's your ale yeast. If it doesn't grow, it's your lager yeast. You had your lager yeast. So very, very simple. From the, let's say, tanks, from the cylindro-conical vessels to the bright beer tank, a few things to look at. pH. Is the pH during the fermentation, is it dropping as expected? And again, when I say as expected, I mean, well, compare it to a baseline that you have already established. So you have already your curves of what it should look like. And if it's like this, then okay, I'm good. I can move on and do some other testing. What about during maturation? Is it moving in any direction? Is it going down? Is it going up? If it's going up, what does it tell you? Maybe I have too much yeast carryover and now the yeast is dying and it's spilling out all its innard. And if it's going down, well, maybe I have some bacteria down there that produces a lot of lactic acid or something. So is it moving? Is it moving based on your baseline? Yeast count during fermentation. Again, you've created your curves. And you take a sample and say, yeah, okay, this is where it's supposed to be, so I'm good, I'm in good shape. And then you can do some HLP screening, just to see if you have any lactic acid bacteria. Of course, the lactic acid bacteria, they're the most prevalent spoilers. They are anaerobe, but they are anaerobe from what we call aerotolerant to facultative. And the difference is that aerotolerant, if there's some oxygen, I couldn't care less, I'm still going to respire, okay? Whereas the facultative anaerobe, they can actually, so aerotolerant cannot use oxygen to grow, and the facultative anaerobe, with or without oxygen, I'm happy. So that's a little bit troublesome, if you will. And of course, they can cause over-attenuation, turbidity, ropiness, and of course, some diacetyl. The HSU, Lactobacillus pediococcus medium, or the HLP, it's very easy to prepare. So I won't go through this, but it's very easy to prepare, very easy to use, and the interpretation is actually fairly easy. Okay, within about 2 days, you can see something, and call it 5 to 7 days, you can be a little bit more sure about if you're dealing with Lactobacillus or pediococcus. So the lacto, and I have a picture, but lacto will look like an inverted teardrop, whereas the pedio will look like a comet. It looks like sesame seeds, or actually a teardrop. And it looks, in a perfect world, this is what it looks like, so that's why I have a little note on the ear that says, description is a little bit subjective. This is in a perfect world, this is what they're going to look like. So those inverted teardrop or those sesame seeds look alike. In the packaged products, well, of course, we have all the lactic acid bacteria, but we have also the acetic acid bacteria. And like I said earlier, they are bacteria that will grow. They are aerobe, so they will not grow in properly packaged beer. But it can, if there's any air ingress, okay, I'm thinking about maybe in a keg or definitely your draft line, that kind of stuff. This is where they're going to thrive. And they're gonna take your ethanol and they can oxidize that to acetic acid, and Acetobacter can even take the acetic acid and go further to water and carbon dioxide. Gluconobacter doesn't do that because it doesn't have the set of enzymes to go to water and carbon dioxide, but it's considered to be a little bit more dangerous than Acetobacter because it doesn't need as much oxygen than Acetobacter. And of course, we have our wild yeast, Saccharomyces or non-Saccharomyces type. The Saccharomyces wild yeast, they're a little bit more difficult to differentiate because they look like our culture yeast. We have Saccharomyces diastaticus. You know what that guy can do? It can actually secrete some glucoamylase and then break down the dextrin, the non-fermentable that was left behind by your yeast, and then slowly but surely produce pressure in your package, you know, increase the level of CO2, et cetera, and we've heard a lot of story about exploding containers. Well, diastaticus is one of them. So diastaticus can produce superattenuation and also has the puff genes, the phenol off-flavor genes that gives you a kind of a medicinal, clove, spicy character that is not really interesting. Then how to detect the diastaticus? There's not yet something that is 100% working and working well and working fast. Even if you look at the STA1 gene, which is the gene that allows that has the glucoamylase connection, if you will. Even if you detect it, it's now proven that even if you have it, it doesn't necessarily express itself. So you might be overreacting. So even if you have a qPCR and you're looking at it and, oh, I have the STA1 gene, well, it's not for sure that that particular strain will express that gene. So that's, you can overreact, if you will. There are some selective media like Lin's cupric sulfate medium and Lin's wild yeast medium. Yeah, they work, but they take a long time, and there's a couple of pictures there. But me, what I kind of like, it's still not very quick, but at least it's going to work. It's the Durham tube, and Durham tube basically it's an inverted tube in a sample of your filtered and autoclaved beer. Okay, so you know that you've killed everything, and then you add to it a small sample of something that you think that might be contaminant. So what happens is that and you put that at room temperature, and you look at the presence of gas. So we see it a little bit clearer on the right-hand side. So you see the tube. So the tube is full with that liquid red over there. And then this is just to show you that with the proper indicator, you can also detect acid-producing bacteria. But see what's going to happen on the right-hand side is that as it is creating or producing CO2, so using what is in the environment, it creates CO2. CO2 goes into the tube, push the liquid that was there, and then you can see a bubble. On the left-hand side, this is just to show you four tubes of belle saison. A belle saison is a diastaticus. So four diastaticus on the left-hand side, and the other one was just to compare it to the Windsor. Okay, and you can see the fourth one, the one with the arrow, 10 to the negative 5. It's one cell of diastaticus per 100,000 cells of yeast. And you see this tiny little bubble, and that tells you. That is in 5 days. So in 5 days, you can see that. Okay. So fairly simple, fairly reliable. The non-Saccharomyces, they're a little bit easier to differentiate because they're so different in colonial morphology. They're very different under the microscope, so they're a little bit easier. You can even see sporulation that doesn't exist in the Saccharomyces. Of course, give you some off flavors, some turbidity, etc. And the classic one is Deboromyces, Candida, and Brettanomyces. Okay, control by prevention. Okay, I said it earlier. You cannot eliminate, you can only start to prevent, and I have kind of called them 8 steps or 8 considerations. And the first one is this: accept this fact. Okay, your brewery is a natural ecosystem. You are walking in, your materials are walking in, so they are everywhere. All you have to do is make sure that you keep them under control. Okay, so first, accept that they are everywhere. Second one is, what is your tolerance? Meaning, establish some very clear specification that is based on, or limits, that is based on your own product. What is the level of alcohol of your product? What is the pH of your product? And those are all parameters that influence a little bit your specification. And don't take a template of specification from brewery A and apply it directly to you. Start thinking about your product itself. Okay, what is my pH? Okay, the lower the pH, the better you are in terms of minimizing any outside growth. What is the alcohol? Comes a point where, you know, even bacteria, they're not so tolerant to alcohol level. What is the bitterness level? Do you have any residual sugar? Do you have any residual fermentable because you did on purpose an arrested fermentation, or you added some fruit puree or whatever, whatnot? Okay, so you need to understand that to be able to establish your own limitation. Just a few numbers here. You're going to get diacetyl of flavors with only 10,000 cells per milliliter. 10,000 seems like a lot, but how much are you pitching yeast? You're pitching, like, if you go by a 12-degree Plato, you're going to pitch 12 to 14 million cells per milliliter. Here we're only talking about 10,000 per milliliter to give you enough to give you some off flavors. Cloudy appearance at 10 to the 7th. And that little picture here, it's a colony that is 0.2 millimeter in diameter, and this is about 10 to the 6th cells. So they're small, but they're pretty nasty. And then finally, I like this one because only 2 cells of lactobacillus by milliliter in the proper condition. Let them grow for 5 days, and it's going to give you enough sourness and turbidity to have this little baby looks like that. But don't give beer to your babies. Okay, you got me in. Okay, so one more sample. You cannot sample everything. Okay, you cannot sample everything. So know your critical control points. And again, yours is maybe not necessarily the same as somebody else or as another brewery, but it's really, really important to understand that. Then move further. Okay, so now you have detection. We are not talking about identification, and I'm not going there today, but you know, now that you have detected something, now go a little bit further and try to identify what you have. And there's a very easy scheme of identification that tells you, okay, what am I dealing with? And the more you're going to know about who you're dealing with, the better you're going to be able to find the source and eradicate or control, bring that back down to where it should be. Don't forget COP, cleaning out of place. Okay? It's very important to realize that, CIP, cleaning in place is one thing, but you have also to understand that you need to break down your fermenters and scrub manually your sampling port, your PRV, your dry hopping manhole, and everything. Okay? A little bit of scrubbing never killed anyone. I like this trick too. If you want to check the efficacy of your spray ball, okay, just after a cleaning, just hose your tank from the manhole, and then take some flour, or if you have a D filter, take some D powder, and just throw some D powder or some flour everywhere like this. So it's going to stick to the wall. And then after that, just start a cycle of just rinsing with your spray ball. What do you think is going to happen? If the spray ball was not well designed, or if it's damaged, or if it's plugged, what's going to happen? You're going to look at it and say, oh, there goes a long line of D that was not even rinsed. Easy trick, but it works. Okay. Food from the dead. Well, remember this, that anything that is there, even if it's dead, it's food for the bacteria. So I've seen some breweries that, they have a very good system of foaming everything, okay, and they let it down, but they weren't rinsing anything. So everything was dead, but what it is, it's nutrients for everything else. So make sure that you clean and you rinse everything. You did what? Have very, very clear and strict procedures. Make sure that they're strict in the sense of temperature, concentration, time of cycles, and etc. And make sure that everybody understands what it's supposed to be. And finally, a return to basics. You know what your flavor profile is supposed to be. And so therefore, establish a good taste panel in your brewery. Make sure that people understand what off flavors are. Make sure that they understand what your flavor profile is, and taste every day. This is a good thing. This is kind of fun, but this is also for your survival. Okay. Now I know that you want, or actually I believe that you need to know more than this. And there's a lecture for that. What it is that we, Siebel Institute, with partner with Domens in Germany, under the umbrella of the World Brewing Academy, we have developed a series of lectures that you can actually purchase à la carte, meaning that you don't have to do a huge 2 weeks program or whatnot. You can pick and choose whatever topics you want. It's an approach for us to help you tailor your brewing education to very specific roles. And here I only list some of the lecture that pertains to what we have discussed this morning, but there are many, many more. If you go to the website right now, we have about 65 lectures that you can again pick and choose. And by mid next year, we're going to have about over 100. From anything from raw material to engineering, so pumps, valves, you name it, everything in between. So it's a very, very nice and easy way to tailor a little bit your brewing education. You have access to a monitor to which you can ask questions during the time of the lecture. The lectures are 100% narrated. It's a very, very good product. I'm really proud of it. We've been working at it for a few years. And finally, control by prevention. Total eradication won't happen. If you have any questions, my name is Richard Dubé. I can be contacted at rdubé@lallemant.com. Any questions, comments? We've talked a lot. I talked a lot. I could have talked a little bit longer. Questions? Yes, sir.
What's your position on yeast washing?
Oh, good. This is very interesting.
The question.
So the question is, what is my take or my position on yeast washing? Yeast washing is a band-aid for those who cannot clean their shit, okay? It's as simple as that. Okay, I come from the big, I admit it, I've never homebrewed my life. I come from the big guys. Okay, so I went from university straight to Molson, and I stayed in the industry, and I loved it enough, you know, that I did my IOB. This is just to show you how old it is, but the IOB, the Institute of Brewing instead of the IBD, which is now. But anyway, in those days, every single batch of yeast was washed. Okay, it is a decent approach. However, that being said, when we're talking about craft brewing and the level that we are probably all part of, to me, it is a band-aid to something because you're so afraid of maybe the technique that you have employed to collect your yeast that you need to do this on top of it. It has some positive effect, you know, it cleans the surface of the yeast because the yeast has the tendency of absorbing some hop material during the fermentation, and so therefore kind of change a little bit the permeability of its membrane. No, it's not effective against all contaminants, and at the end of the day, if it's not perfectly done, pH-wise, proper agitation and time, well, what happens is that you end up weakening your yeast. So, I say, clean your stuff, make sure you have the proper harvesting technique, make sure that all your personnel is properly trained, and forego the idea of washing yeast. That's me. Don't need to repeat that. Any other questions? Well, thank you very much for coming.